The discussion, and I hope you have some questions.
Where is the... I talk too much, so people get tired.
First project. So why did you choose because Cyprus, so island population can be isolated?
Yeah.
Can have larger founder effect? I know that there were several migrations.
Yeah, they are not isolated, and they are actually quite the other way around. Well, actually, it's more for practical reasons that mere practical reasons that we looked at Cyprus, because if you look at the region of the Near East, I mean, at least in this moment, it's not the easiest places to go for collaborations. And it was also based on the fact that we could find collaborations with people that were working on those aspects or interested in those aspects. So that was the main decision for taking somewhere in between, because in terms of environmental factors, and this was the latitudinal transect that is in the title of the project. Sorry, the idea is to have something different in environments also, because maybe we're talking about adaptation to the chemical environment independently of culture, or, you know. So the idea was to have some populations around here, and it came out that it was Cyprus. But we know from many other studies that Cyprus is not an isolated population. On the contrary, it was like also used in commercial routes, and so. Sorry?
There were several migration rates.
Yeah, yeah. Yeah, but we have populations that are separated by sufficient distances for differences to pop up if the differences are there. That's the idea. And then we can also compare with populations from other populations for which data, at least genetic data, exists. We won't have this bottom-up approach with the micrococktail, but at least for genetic data, there are the 1,000 genome project. There are a lot of data, so that's the idea, yeah.
I have one more general question. So looking at selective pressures related to diet, so there are some quite number examples, as you were talking first about lactose tolerance. To get more broader knowledge into this area, then I would perhaps look at some specific diet which some populations rely to heavily, and then there might be a way how to see whether there are selective forces for metabolizing such diet. So what would be your guess, let's say within Europe, what was not researched so far, and what would be such a specific diet in, let's say, historically European population? My guess would be, let's say, some population which heavily rely on, let's say, marine fishing, or something like that.
Well, marine fishing, this is something very old, no? The populations relying on fishing.
Heavily relying on fishing, and which might be. Okay, I'm not.
No, it's
metabolism.
Yeah, it's not very easy actually, because we use those broad, rough classifications based on subsistence mode, but actually this is something that we discussed in this paper, that okay, pastoralist populations in the Sahel, they rely a lot on milk, but they also have exchanges with there is all a network of exchanges with agriculturalist populations that give them cereals. So they also rely on cereals. So the differences in diet can be quite difficult to show up. Lactose is very popular because it's somehow easy, and indeed in Europe it is clear that up to not that long ago there were populations who were heavily relying on milk at some moments. I'm not sure I understood your question, but for instance for those ADME genes it's more complex because actually these okay, because these are involved in metabolism of toxins. So, for instance, NAT genes acetylation genes are very specialized in some kind of toxins, and it's not easy to show. For NAT2, there have been hypotheses on the fact that you would cook your meat at very high temperatures or not, because at high temperatures you produce these heterocyclic amines and things like that that are metabolized by NAT2. But in a more general way, we are using proxies, so of course it's this classification of populations according to subsistence mode is something that dates already from Mourant and these people. But
that might be another good guess because, based on this latitudinal gradient, you usually see higher proportion of meat in northern diet. So perhaps some of the genes which are related to metabolism, related to smoking or of meat cooking, which seems quite toxic in high concentrations.
Well, NAT2 is one of those genes involved in those things, but it's very difficult to show. I mean, yeah, again, there have been hypotheses, but nobody has managed to show other than the fact that you have differences in the phenotypes. But the causative factor, that was my point, the causative factor, actually we don't know. And we're trying, with this approach, by classifying populations according to different criteria, we're trying to actually head towards the causative factor. But I don't know if I answered your question.
So, I'm actually grateful for very interesting questions.
Thank you. Thank you.
And now, a few words in English and in Czech.
Okay.
It's not large number of students, unfortunately. Yet I have been working as a student's medical doctor. Já jsem pracoval jako lékař mnoho let. At the moment, I would like to express and encourage our students for this
Thank you.
study.
Thank you.
450 and mitochondrial genetic is the top of the population.
Thank you. So Mederick has a promising PhD coming up.
So other question or comment, please?
I'm sorry, I'm one more. There are some discussions related to populations which heavily rely on soya diet, for instance. As soya, consumption of soya is related to phytoestradiol, and it is thought that in these populations there are differences in estradiol receptors, and I'm actually not very familiar with this.
Me neither. No, but finish. Go ahead. I don't know this.
How far or how settled are these discussions?
Oh, I really cannot help you on that, because I'm really not specialized in differences in diets between populations. That can be a topic that Médéric will have to to deal with precisely in his PhD. But is it soya consumption is heavily relying on soya consumption? Is it a recent phenomenon or is it a traditional phenomenon?
Like in Eastern Asia, that's quite.
But since traditional, okay. So.
And the soya contain a lot of phytoestradiol.
Yeah.
Then it is thought that this is related to higher frequencies in different receptors than we can, for instance, find in Europe or other parts of the world.
Yeah. I don't know. Yeah, at one moment this is a little bit off the microphone. We tried to set up a collaboration with a colleague somewhere in the world, I won't say where, but where there are populations that are traditionally vegetarian and populations that are traditionally non-vegetarian. And the idea was to compare an NAT2 evolution in these two evolution or diversity in these two kinds of traditional dietary cultures, but unfortunately the collaboration didn't go very well, and somehow he did some study without without collaborating, and he didn't do it very nicely, so I'm not sure that his results were finally very no, I don't think that his results were how do you say, founded? No, solid or whatever. But these are kind of directions that can be taken. Yeah.
So I have one quick question related to NAT2 polymorphism. So it surprised me that it's still so polymorphic. Okay, you showed us the change in the percentage of allele frequency across populations. However, still, if it is under stronger selection and taking into account this strong founder effect across human populations of human beings across the world, I'm still surprised that there is such a big polymorphism in nearly each population. And
well, the idea.
I don't know if it is really so adaptive, or it can be adaptive, but maybe it's not so easy. It can be in some, say, epistatic interaction, which would create the slow testing, slow acetylators when it's gray.
Well, maybe not of course, there can always be epistatic interactions, but in this case, really, it's those polymorphisms within the exon which are shown to. But I think that you are right. Of course not. Of course not. Of course not. And one of the ideas of projects like the one that we want to do here now in the next days is to be able to really correlate genotypes with phenotypes. But in this case for NAT2, that's a thing that we discuss also, that okay, you have all those slow variants. If the ancestral form is the fast acetylation, somehow these slow variants are slightly deleterious, somehow loss of function alleles. So it can also be that there has been some relaxation of selective constraints, of functional constraints on the gene, that the gene has become less important, and so he's able to accumulate all this diversity, of this detrimental diversity. But there is still the fact that you find this in particular modes of subsistence and not in others. I mean, in hunter-gatherers, you have really much less diversity of slow haplotypes, much more fast. Yeah, of course, they drift.
Agriculture, there was a population growth, so which would take up the slightly diversification.
Well, no, drift would they would drift less, so that these variants, many of which might be quite new, that they become so frequent, it's difficult to be explained by drift. On the contrary, so yeah, it's not conclusive, of course. These are open directions.
Nicely forms the genomic variation. It is less variability
No, this is not variability. This is only the phenotypes. Huh? So I don't have it here. These are the phenotypes. I don't have a slide here, but actually you have. I have a slide here. If I put my key, I can show you a frequency variation in the world. But I should put my key. But I mean, you find. With the sample sizes used today to determine the polymorphism, but you find between 6 and 10, 12 haplotypes in almost all populations. From 6 to 12 NAT2 haplotypes are detected in most regions of the world. Maybe not in America, maybe not in Oceania, although I don't have it in mind, but yeah, I should watch a slide to let you know. But this is only the phenotypes.
So only here was the polymorphism loss, and otherwise.
Well, no, because this can be several fast acetylators. This is only the predicted phenotype, so it's a combination of haplotypes. So maybe here, but I cannot tell you by memory what there was there. But these are also, in general, small sample sizes, small studies. But it can be 2 or 3 fast haplotypes that give a frequency of almost 100%. Yeah, I have a slide, but I should move on. This is the predicted phenotype proportion.
But still there is like...
There is not this trend. Really, there is no correlation of genetic distance with geographic distance for NAT2. There is no correlation of loss of diversity, at least with the data at hand, of loss of diversity with distance from Africa. And there is really this pattern that is quite unusual of East Asian populations, quite differently from European, sub-Saharan African populations. This goes all the way down to South Africa and all the way up to Western Europe. Huh? And it's mainly in East Asia that there are big differences. You're welcome.
So, any other questions, please? Last chance for question. If not, thank you very much.
You're welcome. Thank you. Thank you.